Hi, my name is Abby Lowenkron, and the project I worked on is called Stability of Evolving Populations in Variable, Heterogeneous Environments and Significance for Origins of Life Studies. We used Python to create a model to better understand how ancient cells evolved into what we have today. The model was a closed system with a food supply, and inside this system were two populations of cells. One population did not evolve, but the other changed from generation to generation, developing different requirements in response to the environment. We observed the growth of the cells in an environment with rapidly changing conditions to observe how the survival of the evolving and non-evolving populations compared. The question we sought to answer for this project was, under which environmental and internal conditions is an evolving population of cells likely to outcompete a non-evolving population? We produced graphs to compare the populations of evolving and non-evolving cells after a set amount of time during which the environment was changing. Altering various parameters changed the output of the graphs. The top graph shows the evolving population in green and the non-evolving population in black. The bottom graph shows P as it oscillates between extremes. I studied three parameters: PO amplitude, or the amplitude of P, the environmental parameter that can represent anything from pH to temperature to salinity, V, representing the volume at which the cells divide, and GrowVF, representing the factor for the volume growth of cells. Our final graphs look like this, comparing the ratio of evolving cells, N2, to non-evolving cells, N1, at the end of each iteration as the variable changes. For PO amplitude, I found that an increase in amplitude did not favor the evolving population. The hope for this study is that in the future it could be continued with a second model that involves different compartments between which the cells can migrate to observe changes over space as well as time. Thank you for listening.